So I put the recording on, and I'm going to share the PowerPoint that I prepared. And in case the recording works, so anyone who's watching this, if you can hear me, that's great. If not, I'll probably upload the morning session, even though we still have technical issues. But on these slides, you have better answers. So I'm going to just jump through the objectives because I didn't update those. But today, what I really wanted to go through was the questions that come up in quiz. And in the morning session, I had 2 questions, especially about the Rh positive and Rh negative individuals and blood types. That is something that 2 students were really nervous about, and I don't want you to be nervous about anything. I'm not asking you to know it super deep. And the quiz questions are also keeping it just very general. So don't let yourself get too worried about it. We're sticking to the very basics of that. We didn't have time to talk about that really in the class. I just mentioned. On this week's quiz, there's actually 55 questions. I normally try to keep it on 5, but because 2 students had started it, I didn't go and remove. I thought that that'd be mean. I might just give everyone 5 extra points or something. I'll figure it out as we go along. And then we'll be doing the lab together tomorrow. And provided that I have all the materials, and so far it looks like I will, you will be actually doing your own blood type. You will find out what blood type you are based on the tests that they would use also in a clinical practice. Those tests are a little bit more automated in those settings, but that will give you a chance to see how the blood type is determined and then think these blood type matches donor and recipient. I won't go through about the practice anatomy at all. That's really quite straightforward, especially on this chapter. It's a lot of cells, looking at them, recognizing. Remember that you can have all your resources with you while you're taking the practice anatomy, and also when you take the test when we get that far. You're able to hear me okay? I assume that you would wave me out of my skirt. Yeah. And if you have any questions at any stage, you just shout out. I want this session to be what helps you. It's nice if it helps other students, but you're here, so it's all about you.
Okay.
And I'm going through every single question on the quiz, and I prepared these slides so that they answer to every single question. And I hope you will see that it's not as hard or as intimidating as the endocrine system quiz was. There was a lot on that. I think this is a lot more, this should be a lot more basic. Because we went through this handout in the class so fast, I just wanted to also show it to anyone who's present here. But if you feel that you have all the answers for it, we're not going to spend time on that. But if you were to say that, no, I didn't get some, I would take the time to go through it. But I'm assuming, since you're looking quite happy, that you got all the answers. Perfect. So for anyone who missed, if this recording now works out, it is available, so you can look at it also on the Canvas. I uploaded the actual one with the answers. I normally try not to upload these because these are all my own drawings and stuff, and I eventually ended up finding those online, like shared, and I was like, oh, maybe I shouldn't be giving all the files. I'll just give printouts. So I'll do it this week, and in future weeks, I'll have a look if I just give the printed versions. So the quiz questions, they're really simple. There's not tough questions. One of the questions, for example, is that what are the parts that make up a circulatory system? Well, circulatory system, as we talked, is made of the heart, which is the pump, and then the tubing, which are the blood vessels, and then the blood that's within those heart and the tubing. So those 3 parts make up the circulatory system: blood, heart, and blood vessels. And that's the very concept that we started our face-to-face class with. Most of the questions on the quiz are from the face-to-face class. There are two mainly clinical questions that are not things that we have time to cover. So I will cover them here and now, or you can use your textbook to find answers. Remember, quiz is not in my class to test how much you know. Quiz is a way of learning things as you're doing that as well. So don't feel that, oh, I should know all this. That's absolutely not the case. The other thing that we talked right at the beginning of the class, which is again a question that is in the quiz, that what are the functions of the cardiovascular system? And on that quiz question, you will be given the functions are transportation, regulation, and protection. And you are asked to drag these things under right heading. So you know that oxygen transportation and carbon dioxide transportation go under the transportation. So we carry oxygen to the tissues from the lungs, and we carry carbon dioxide from the tissues back to the lungs so we can breathe that away. So those are both examples of transportation in the that the blood plays role. We also have hormones being transported by blood, same as nutrients. And one thing that I didn't really say in this form, I just said waste products. But an example of waste products would be urea. And urea is what's circulating on your blood until it gets eliminated by the kidneys. So if you've ever met a person whose kidneys are not working quite well, their eyes, the whites of their eyes are very yellow. So that's because there's a high amount of urea in their blood, and that actually shows up there. So the color of the blood can be used there. Color of the blood showing through the eye whites can be used there to tell that something's wrong. So all of those things should go under transportation in that quiz question. The two things that you have on that list on the quiz that go under regulation would be pH, and especially the buffers that we use with managing the blood pH. And we remember that the blood pH was between 7.35 and 7.45. You don't need to memorize that by heart, I think, as long as you recognize that number. But this is maintained mainly thanks to buffers. And I don't know, since I didn't teach your BIO 181 or BIO 156, but in my BIO 181, BIO 156, we actually do a whole lab. We spend a whole week looking at what things are good buffers, what are not good buffers. So buffers are chemicals that help to keep the pH within a narrow range. Another thing on that quiz that goes under regulation has to do with temperature regulation. And the quiz goes a little further. It uses terms vasoconstriction and vasodilation. Vasoconstriction means simply the blood vessels being made smaller, and vasodilation, the blood vessels diameter being made larger. And by constricting and dilating, we were able to direct blood either to the extremities or keep it away from there. So if we needed to lose heat, we direct the blood there, and then we hope that with sweat and evaporation and so on, we get rid of it. If we need to preserve heat, we stop blood circulation to the extremities and really reserve the heat that gets transported with the blood only for the vital organs. There were two things on the quiz as well that go under the protection. So I think maybe three things. So blood helps us to protect us against pathogens, against infectious agents, and it also protects us against bleeding. And that was the platelets, the function of platelets. They helped us to form a clot or a scab or I'm thinking what other all sorts of names we can have for it. And we'll talk about the platelets and the scab formation a little bit today. The other thing, these numbers are something that you are being asked to identify in the quiz. And I think we really only talked about, oh, I didn't mean to do that. Sorry, we only really talked about the pH in great amount. I might have mentioned the total blood volume, but we didn't really dwell into it any further. So the pH was somewhere between 7.35, 7.45. There's about 5 liters of blood in an average individual. This varies a little bit. Generally, males are a little larger than females, so males tend to have a little more. It's about 8% of our body weight that we have blood. So for females, between 4 and 5 liters, for males, between 5 and 6. Osmolarity and white blood cell count, I know that we definitely did not talk. So a typical white blood cell count is somewhere between 5 and 10,000 cells per microliter, and osmolarity between 280 and 296. At this point, you don't need to worry about those, but since it is a question in the quiz, I wanted to give you those numbers so you can pick them. And if I'm moving too fast or too slow, just let me know. Then I wanted to talk a little bit about plasma. We talked how it's a sticky straw-colored substance. The quiz does ask you that how much of plasma is made of water, and I think I said 90%, it can be 92%, around 90% number by weight of the plasma is water. The most common protein that we end up finding in plasma is albumin. 60% of all the proteins in plasma are albumin. So both of those I do remember very clearly were asked in the quiz, that you can tell that it's 92% water and 60% albumin makes albumin the most common protein to be found in the plasma. An example of the waste product that we find in the plasma, urea would be an example of that. And that's really the most common waste product and most common nitrogenous waste product that we see. Again, don't let that term throw you off. We talked about that, but we just didn't talk with the as fancy words. So those are all the questions that I could think of the plasma in general level. There were 2 other that I wanted to kind of highlight. On question, there is a question where you compare plasma against formed elements. And you remember that it was the formed elements were all the cells within the blood. So that was the red blood cells, white blood cells, and platelets. So you will be given a list of all of these things that you see on the screen and asked, would they be found either in plasma or would they be found in formed elements of the blood? Well, in terms of the plasma, we find a lot of proteins there. The albumin was the one that I mentioned earlier on, but also globulin and fibrinogen. They are listed there on the quiz as things that you can find in the plasma. Other things that you can find in the plasma were nutrients, and I didn't use the example of glucose, but glucose would be a good example of nutrients that circulate within the plasma. And that is listed there on the quiz as they ask you to make sure that you know that that's part of things that you find in the plasma. We also have talked quite a bit when we talked about blood transfusion about antibodies. So antibodies are also found in the plasma. You find hormones, and one of the chemicals that you can find in plasma is chloride. So that one we didn't mention. That is just one of the things that you need to be able to drag to plasma, the chloride. So that might be something that you take a note if you didn't get a chance to do so. When we looked at the formed elements, well, this makes our life easy because we went through these quite well. So red blood cells are one of the formed elements. They just happen to use the fancy word for red blood cells in the quiz, so erythrocytes. Same goes for white blood cells. So rather than saying white blood cells, they ask in the quiz that you would recognize that leukocytes are rather found in formed elements than in plasma. And then all of the white blood cell types, I don't think they asked all of those, but they asked you to recognize a couple of those and be able to track them under formed elements. So you remember neutrophils, leukocytes, monocytes, eosinophils, and basophils. Those were all examples of white blood cells. And I think they were on that handout that I gave, but again, remember every way that you can use, you're allowed to use both in the tests and with the homework, your notes, everything. We also talked about platelets being part of the formed elements. So that would be a good one. The neutrophils, I think that's the white blood cell that they asked, the type of white blood cell. So I just listed it twice there. I think they also asked the monocytes. So both of those are examples of white blood cells. So they're found in the formed elements of the blood. We should talk a little bit how formed elements are produced, since it comes up in the quiz. Formed elements are produced in red bone marrow. The production of the formed elements in the red bone marrow is known as myeloid hemopoiesis. In addition, some of the formed elements go to other parts of the body, in addition to bone marrow, to mature and develop further. These parts where they would go are known as our lymphatic tissues and organs. And when they go, these white blood cells, to lymphatic tissues and organs, we talk about lymphoid hemopoiesis. And really, I think that this question, the reason why I put it on here was that the quiz question was a true or false statement, and they called red bone marrow, production of formed elements in the red bone marrow, lymphoid hemopoiesis, and they called production at the lymphatic tissues myeloid hemopoiesis. So they had mixed those up. So you needed to spot that that statement was wrong. I think that was the trick on that true and false question. So don't fall for it. Even though everything is correct, just make sure that they use right terms for right things. In terms of the formed elements, we are going to go through those once more because there's many ways that they get asked in the quiz. They get asked that you are asked to recognize them from pictures. They get asked that you can pick them from lists. They get asked that you can drag them to boxes and so on. So we remember that the most numerous of the formed elements was the red blood cells. And the term that we are going to use when we talk about red blood cells was the erythrocytes. The other question that I saw on the quiz was that it asked, what is the most important function of erythrocytes or red blood cells? Well, the transport of respiratory gases would be the answer to that. I don't think that the question is totally fair because we talked about the regulatory and protective functions as well. It's not just about transportation, but what they are looking on the quiz is the transport of respiratory gases. And we remember all what the red blood cells look like. They were those flattened discs. Then you are going to be asked to identify white blood cells so you can drag them name to the right picture. So remember that white blood cells, we rather use term leukocytes when we talk about them. And the neutrophils were these white blood cells that had a significant role in infections. Lymphocytes, more to do with antibodies, memory cells, destroying any virus cells or cancerous cells. So we end up having a lot of cells that become cancerous throughout our life. But lymphocytes are really good and they catch them before these cancerous cells get to multiply and spread anywhere. So cancers do happen, but your body typically is able to catch that. Same as if a virus enters to your cell and turns the cell to do something that cell normally wouldn't do, such as becoming a factory or a machinery for making more viruses. These lymphocytes detect that and go and destroy these cells. Monocytes are focused on the immune clearance. So dendritic cells and macrophages are examples of monocytes, and eosinophils were to do with parasites. I think they gave a particular parasite that you were supposed to be able to tell that, oh, eosinophil would deal with that. And I think that that's on one of my last slides for this review of the quiz. Basophils are associated with inflammatory responses. So those are the white blood cells. There's the pictures, and it's the exact same pictures, if I remember correctly, that they actually use on the quiz for recognizing those. So whether it's a screenshot or whatever helps you to recognize those, that's the way to go. You have very good notes also on your textbook and so on. The final element of the formed elements that I want to talk about real briefly is the platelets. And you might remember that I told platelets being cell fragments, and their role is to be clotting factors that then give rise to the blood clot formation. And we'll talk about that process a little bit more than we had a chance in the class. One thing that I do think was a small question in the quiz, but a question that could be tricky. Remember that white blood cells spend only 2 hours in the bloodstream. They actually don't circulate in the bloodstream. They end up traveling to your lymphoid organs, being stored there and recruited when needed. So they really quickly leave the blood after 2 hours and cross those blood vessel walls going into the tissues. There were probably 4 or 5 questions out of those 55 questions where you were asked to put things in the order of most common to least common. So that's something that they love to ask on the quiz. So we remember that red blood cells, so our erythrocytes, were the most common one. There's about 45% of those, or the 45% of the blood volume is red blood cells, whereas platelets were less than 1% of the blood volume. White blood cells are also less than 1%, but they will ask you to list each different type of the white blood cell as well in the order how common they are. I'm just making a note so I know to give extra credit for anyone who attended today. And you remember that the order for remembering what's the most common and what's the least common of the white blood cells, well, the mnemonic "never let monkeys eat bananas" work very well there. And like I said, there were many questions where you were asked to put these in order. So it's good to have that somewhere on your notes. In terms of the red blood cells, we've already talked about, make sure you recognize the term erythrocyte, referring to the red blood cell. One thing that there were few questions was about the fact that there is no nucleus in the red blood cell. And because it has no nucleus, red blood cells are not able to go through mitosis or make any protein. If you remember when you learned about cells and what cells do, if there's no nucleus, that was the brains, if you wish, of the cell, you needed to have the DNA to be able to go through mitosis. And the DNA was used also to make proteins, kind of as a recipe for how to make up a protein. If you don't have any of that, you're not able to do that. So red blood cells cannot go through mitosis. They cannot make proteins. The other thing that the quiz kind of touches upon, that's worth reminding ourselves of, is that there were no cell organelles in red blood cells other than just the nucleus and DNA, and one of those was mitochondria. You remember that the mitochondria is often described as the cellular powerhouse. Well, if you don't have mitochondria, you cannot do aerobic respiration. So that's why the red blood cell does not end up using the oxygen that it's supposed to carry from one place to another, because it doesn't have these cell organelles that would then consume the oxygen. The main function of the red blood cell, like I said, that was a question on the quiz. The main function of the red blood cell is the transportation of oxygen from the lungs to tissues and carbon dioxide from tissues to lungs. So be familiar with that. And it does ask the quiz that you are able to recognize that there are 4 heme groups in each hemoglobin. And because there are 4 heme groups, you are able to transport 4 oxygen atoms on one hemoglobin. So that's worth knowing the number 4. They give you a bunch of different numbers, and you're supposed to be able to pick that. Let me know if you have any questions. Yes.
I have a question. Do you have to label anything else in the hemoglobin, or is that it, just the 4?
No, you don't need to label anything from the hemoglobin. You just need to be able to say from hemoglobin that there's 4 heme groups. Nothing to label in the quiz. That was a very good question. The one point that it does say, and I just thought that that might help you when you're tackling the quiz, 33% of the cytoplasm of a red blood cell is made of hemoglobin. So that might be a note as well. It might help you to pick the answer hemoglobin when you remember that there's 33% of the cytoplasm is made by the hemoglobin. Well, in terms of the white blood cells, it will ask you the quiz to list them, like I said, in order of how common they are, order of abundance, how abundant they are relatively to each other. So the order is neutrophils, lymphocytes, monocytes, eosinophils, and basophils. And that order, the mnemonic is written there, but you can come up with other. I've seen some other better ones. My mnemonics, I try to keep the family-friendly ones on these classes. Yes.
All right. Are we going to have to know like percentages too? Or
No,
just?
no, you don't need to know percentages. You just need to know what's the most common, what's the least common, and then what's in between. Perfect. And actually on the exam, I don't even ask you to know how to write those, as long as you're able to pick them from a list. On the practical exam, you are asked to write out the name of the white blood cells. But remember, you can use your notes, and I give points even if the spelling is wrong. The automatic grading will look something, but then I will go and give you the points for those.
Okay.
Perfect. One thing that I'm hoping that the annotation works. One thing that the quiz talks quite a lot about is the production of different kinds of blood cells. And the production of the red blood cells, you remember, that was called erythropoiesis. Oh, don't worry. I had a lot of trouble. I just got a chat message that there's a little bit of trouble getting the audio working. I had a lot of trouble, so don't worry. It's probably something bigger. Actually, I spent good half an hour in another class fighting with that. So let me just quickly put that to the chat so she doesn't worry. So, like I said, the quiz asks quite a bit about the production of different blood cells. So remember that erythropoiesis refer to the production of the red blood cells. And the steps of production are pretty much the same, regardless of what type of cells we're going to be looking at. So everything starts with the stem cells. That's the start of production of any type of blood cells. Sometimes they're hemopoietic, sometimes they're pluripotent stem cells, but they are stem cells of some kind. And you remember the stem cells is the cells that can become any type of cells at all. So they are not yet locked that they need to continue to become a certain kind of cells. And I will just answer to the next step in the production of the red blood cells is that from the stem cells, they become colony-forming units. What colony-forming unit means that these cells now have receptors for factors that stimulate their development to become a specific type of a blood cell. And the third step is precursor cells. And precursor cells, there's two types that we are going to be familiar with: erythroblast and reticulocytes. And these then eventually mature and become red blood cells, mature erythrocytes. There is going to be a question where you are going to be asked also to match these pictures with these steps, but there is the name of the cell type underneath that picture. So the first one would say that it's a stem cell. Second one would say that it's a colony-forming unit. The third, there's two pictures for that. The erythroblast, which is the first one that we see here, and the reticulocyte, which we see as the second one here. And of course, the red blood cell, the mature erythrocyte. So as long as you're able to tell that that's the order of making red blood cells, you're going to be just fine. It's not going to go into great detail. In terms of the hemoglobin that we had in the red blood cell, there's going to be a diagram like this. We're adding a little bit of data reading skills on our quizzes or critical thinking skills. So we're giving you a diagram and asking you to see what's happening there. So you saw that hemoglobin was broken into heme and globin when it gets taken off from the blood circulation. So we break it into its components so we can reuse most of these components. So there's going to be a picture where instead of heme, there's a yellow box, and if I remember correctly, number 1 in there. And you are asked that what should go into that yellow box. And what should go there is the heme. You'll be given a list of options, but what should go there is the heme. In terms of the leukopoiesis, so production of the white blood cells, it's very similar to what we did when we went through the production of the red blood cells. So everything starts again with stem cells. In this question on the quiz, you're asked to organize them in order, that choose 1, 2, 3, 4, that which is the first step. First step is the hemopoietic stem cells. So stem cells could become any type of cell. So that's why we start with those. The next step was the colony-forming unit. Colony-forming units have receptors for factors that stimulate the development. Then they become precursor cells. And I listed 3 different kind of precursor cells that we find with white blood cells. I don't think it asked anything about those, but I listed them just in case anyone needs more information. And final stage was the mature cell, mature white blood cell that we ended up with. So just knowing that order of the elements in the production would be handy. Final production, production of the clot. There will be a picture on the quiz, which is this exact picture. And you're being asked to drag the proper labels to the proper spots. So we're going to go through all of these and make sure that you're able to do that and you're recognizing what's going on. What I do want to highlight here is that what we're seeing is, I'm trying to move the boxes so I see the screen as well fully. What you will see is bone cavity or bone. This all is inside the bone, what we're seeing here. This all is inside the bone. So this is a sinusoid of bone marrow. So bone marrow, the empty space within the bone marrow, what we're looking here, the inside the tube or inside the vessel that we see there. And the cells that form the lining for this sinusoid of bone marrow are known as endothelium. So endothelium is the lining of the cells that form that are around the bone marrow. So if you're able to tell apart those two, you're in a great place. Then what we are talking in that picture, or what you are asked to notice, is also to recognize that what we're seeing here, this big cell that has parts of it extending into the bone marrow. This big cell is a megakaryocyte. Megakaryocyte is a cell that gives off or sheds platelets into the bone marrow. So those small cell fragments that platelets are, they come from megakaryocytes. And before the platelets become platelets, when they are shed by the megakaryocyte, they are first proplatelets. So proplatelets are these bigger than a single dot. They are an intermediate, so kind of middle stage between the megakaryocyte and a platelet. And then they become platelets when they have matured. And platelets were important, helping with the blood clotting. So if you're able to spot those, you're doing amazing. The two other ones that I think that are familiar from things that we've seen, we should be quite happy to spot the erythrocyte, so the red blood cell. It's going to ask you to be able to identify that, and it's going to ask you to be able to identify white blood cell. And those are there on the side. So it literally is this slide. So if you want to take a screenshot, whatever helps you, or if you want to look at the recording, it is going to be available for you. A couple of questions that we have about platelets. I think there's a choose all options that apply question, and these all are true statements about platelets. So platelets stick together to form a temporary platelet block that seals any breaks in the blood vessel walls. They take in and destroy bacteria. They also secrete growth factors that stimulate mitosis in fibroblasts and smooth muscles, which then again helps to maintain and repair blood vessels. And finally, platelets initiate the formation of clot-dissolving enzyme. So this dissolves blood clots that have lasted beyond their need. So these blood clots are now still existing even though they're no longer needed. So platelets secrete enzymes to break those apart as well. So just being able to spot that those are all true statements about platelets would come in super, super, super handy. So I think it's a list of questions, and it says that pick all that are correct. So if you're able to pick those, you're doing great. I'm just not going to rush so you get a chance to take your full notes, so don't worry. You just wait me when you're done, because it's a very wordy way how I've written it.
It is.
I really appreciate it. I would be surprised if you don't do extremely well. You really are doing so much work for this class.
I hope so.
I can promise that I will recognize it in terms of the extra credits. Your work effort has been fantastic.
Thank you.
You'll find out that I'm all about extra credit in my classes. Like you can get extra credit for the most random things.
For bringing donuts.
You don't have to bring donuts out, but you have to eat donuts when I bring. No, you don't have to eat them, but I really do appreciate people who show up to the class and who put in the effort. Really, in my mind, it's all about putting in the effort. It doesn't even have to be about getting everything. A-grade student doesn't have to get everything correct. They just have to try. So
Try to.
as long as you answer to a question, I always give points if you answer to a question, even if it's a wrong answer. At least you've tried.
That's the best way. Okay,
Good.
I finished.
Perfect. Thank you. I know it was a lot. The other two terms that come up in the quiz is going to ask, what is fibrin? So fibrin creates the framework for the blood clot. If you're able to spot that when it asks that, what is it out of the following ones that creates the framework for a blood clot, the correct answer for that would be fibrin. I think there's also a question where it asks that, where do we see that or why do the healthy blood vessels, why don't they end up developing clots on those? So healthy blood vessels, their walls don't end up developing clots all of a sudden, and that's because platelets are not able to attach to the wall of the blood vessels because these walls are coated with prostacyclin. So that's a term that it gives you a list again and asks you to pick that which one of these is the one that lines or coats the walls, the endothelial walls of the blood vessels so that the platelets are not attaching to those walls in healthy blood vessels. So that would be a good term to be able to recognize.
Oh,
you got
you
it.
can go.
Excellent. This next question, I've taken completely copy-pasted from the quiz because this is something that we didn't talk about in the class and we didn't really have time. And what I'm asking there, or what you're being asked, is to recognize terms. So what is important there to see is that plasminogen gets converted into plasmin. That's really what the question is asking. It's an extremely wordy way of asking that what gets converted into, or what plasminogen gets converted into. And it is the plasmin that it gets converted into. It's just the details of the blood clot formation. So there will be a sentence where there's that word missing, the plasmin, and you just need to be able to pick it up from the list of the options that it gives to you. There are two questions about the blood clot formation, including this one, where you have pictures of different stages of how blood clot or a scab or a plug gets formed. So what we're seeing on the first picture here is that the blood vessel wall has broken here. You'll notice how the wall of the blood vessel is here broken. You see that the blood would just keep flowing out of there if we don't do anything. Well, the very first thing that your body will do to stop you from bleeding to death is that your blood vessels will have something known as vascular spasm. It means that the blood vessels will constrict. They try to make the diameter of the blood vessel as small as possible so we don't end up carrying a lot of blood to that area that's bleeding. So we simply want to make the blood vessel small. That's what the vascular spasm is. The second stage of platelet plug formation is the platelet plug formation, or the second step of the hemostasis is the platelet plug formation. And in that, you can see that we are now attaching to this collagen that's visible outside the blood vessel wall. That's an important thing to notice, that the endothelial collagen exposure is what really drives the platelet plug formation. So those are the three terms or three statements that I would match with the platelet plug formation. And final stage is coagulation, and that is the fancy word for blood clot or clotting. And what happens there? Fibrinogen gets converted into fibrin, and there's both intrinsic and extrinsic mechanisms that are involved in there. And we're going to talk about those intrinsic and extrinsic mechanisms more, but you don't need to know a lot about them. Just know that you need to know the very basics. So, blood clot can happen in two ways to start with. Extrinsic mechanisms refer to when you have something, and I'm going to jump actually to here first. Extrinsic mechanisms are initiated by clotting factors that are from the blood vessel wall or tissues around the blood vessels. So it's not the blood itself, it's things other than the blood that are extrinsic mechanisms of the blood clotting. Intrinsic blood clotting mechanisms have everything to do with factors found in the blood itself. And then after a couple of steps, these two fuse together and we have a common pathway of blood clotting. But remember, intrinsic, it's all to do with things that we find in the blood itself, and extrinsic has all to do with things other than what is found in the blood. So for extrinsic, the damage to the vascular tissue or tissue around the blood vessel would be an example of that. Factor 3 and factor 7 are examples of extrinsic. All other factors, factor 12, factor 9, and factor 11 are examples of intrinsic. And then the common pathway includes the protothrombin activator and fibrin cross-linking, as well as the factors 3 and factor 13. I had to do some math there. The Roman numerals are not my game. So as long as you're able to recognize those. Then there is a question that how does your body prevent clots from happening when you're normal and healthy? Well, there's three ways how that happens. Repulsion refers that when blood vessels are not damaged, the prostacyclin coats the blood vessel walls. And we saw the prostacyclin as the thing that prevented the platelets from attaching to the blood vessel walls. When everything's going fine, when blood vessels are undamaged, everything's good. So we prevent from any clots forming by this method. The other method how we prevent blood clots from happening is through dilution. In dilution, we have a normal amount of blood flowing through that area all the time. So just by having that normal amount of flowing, we don't end up with any clots forming. Issue becomes if for some reason the normal amount of blood gets disturbed, there's not as much blood flowing as it should be through that spot. For example, in a circulatory shock, all of a sudden we have less blood flowing and we are more likely to start forming the blood clots. Blood really, if you leave it on its own, it's not moving anywhere, it tends to start to clot. That's what happens with nosebleed. If any blood falls on a table, it soon becomes hard. That's because of this clotting happening automatically. Anticoagulants are the last method of clot prevention. So when you have antithrombin, that causes the thrombin production to go down. The other one that is in the quiz mentioned is a drug called heparin. Heparin is used for heart attacks and untreated angina, and it causes the formation of protothrombin activators to go down. So it depresses the formation of those. It also blocks the action of thrombin and fibrinogen, and it promotes the action of antithrombin. So on that question, you're being asked to drag these, what I have indicated here with arrows, under the correct methods of clotting prevention. So you know what repulsion is, you know what dilution is, you know what anticoagulants are. Really, it's just a matching thing. Did you get those? Am I rushing? No, you're good. Okay. Then there is a question of effects of both the environment and diseases on formed elements. So how formed elements are affected by different environments, by different diseases. Again, you will have headings increases something, it has no effect, or it decreases something. So you are being asked to notice that high altitude promotes red blood cell production. So when you're in a high altitude, you make more red blood cells. We talked about that, how athletes go to train on high altitude before competitions, for example. If you have little oxygen in your body, that's known as hypoxia. If that happens for a long time, that will also cause for your red blood cell production to go up. So your body is trying to respond to the fact that we have a small amount of oxygen in the body by making more red blood cells. Another thing that can cause white blood cells this time to be produced is if you have any kind of infection. And the question that you will see on the quiz is about influenza infection. And the type of white blood cells that are especially produced in response to influenza are B lymphocytes. We have not talked about B lymphocytes yet. We will talk about them when we get to the immune system. But at this point, as long as you're able to spot that there's a statement that influenza infection causes B lymphocytes to be produced. If you have a chronic asthma, that causes also certain white blood cells to be produced more. And the type of white blood cells that are especially relevant with asthmatic responses were the basophils. So I think that the question in the quiz doesn't really say white blood cells, it just says basophils. So you're supposed to know that basophils are an example of white blood cells. And finally, the parasites. If you have a parasite, for example, tapeworm, and that's what the question uses as an example. If you have a tapeworm, to respond to that, your body will start to produce more white blood cells. And the special type of white blood cell that was really good at dealing with the parasites was the eosinophils. They're extremely good at dealing with various different parasites, including tapeworms. The other end of the spectrum is that what kind of things in our environment and various disease situations could cause things to be decreased or depress the production of things. So let's have a look of that. If you do not get enough iron from your diet, so you're not getting enough iron from your diet, dietary iron that you should be getting, because you don't have that iron for red blood cells to be made, the red blood cell production will go down. There's just nothing to make them with. Another example where the red blood cell production goes down is EPO hyposecretion. You remember hypo means that something is done very little. If we have very little EPO in our circulatory system, the production of the red blood cells goes down. There's normally some amount of it. If there's only very little, the red blood cell production decreases. Another thing that can cause red blood cell production to go down is if we don't have a lot of intrinsic factor around. So small amounts, the arrows are here very important. If you don't have a lot of intrinsic factor, the red blood cell production goes down. And finally, if you are bleeding, and that's an acute bleeding, you're losing a lot of blood all at once, as a response to that, you can actually have that the red blood cell production goes down because we're now just simply trying to deal with that loss of blood. We're not replacing it with red blood cells. If you have an acute viral infection, that will not have any effect. The viral infection will not have not positive and not negative effect on the production of the red blood cells. So really, this is a question in the quiz where you just drag those to the right place. I know that we have hit the 9 o'clock mark, and I promised to try to keep this short, but I have a little bit more material. But if you have to go, I totally get it. I will still record it, and it will be available as a recording. I appreciate if you stay, but you don't have to. So I'll just carry on. Ask me if you have any questions. In terms of the blood transfusions, so we're approaching the kind of end of the material that's in the quizzes. The big thing to remember here is that we are going to need to know about ABO typing, really the Rh positive and Rh negative. We will touch upon it, but you don't need to know great amounts. There are over 30 other types of blood, other blood types, but we're not going to worry about them because they are rare to cause any issues with blood transfusions. So it is the ABO types and Rh positive or Rh negative that cause transfusion issues. Other blood types, they don't make as much of an effect on the issues that we might see with blood transfusion. So it's only the ABO and Rh positive, Rh negative that are the ones that we're going to be focusing on in terms of transfusion issues. And that's a question, I think, in the quiz that you recognize that the ones that we're going to focus is the ABO and Rh positive, Rh negative. We talked in the class about this, that you could have a blood group A, B, AB, or O. And if you had a blood group A, that meant that on the surface on your blood cells, you had antigen A's, so sprinkle A's. If you had blood type B on the surface, you had antigen B's, sprinkle B's. On the surface of a blood type AB, you have both A's and B's, and on the surface of the blood type O, there's no antigens there. The quiz is going to show you these 4 types of the antigens on the surface of the red blood cell, and it's going to ask you to recognize that when you have 2 types coming out from it, that's going to be blood type AB. When you have 1 type, it's going to be either A or B. When you have nothing other than the basic stick coming out, that's blood type O. But really, the quiz simply asks you to recognize out of these 4 pictures that which one of those pictures would be likely to be an example of AB. And of course, it's going to be the one where you have the 2 present. In terms of the blood transfusions, what we needed to know that normally all the red blood cells are nicely separated, but if you have antibodies present, remember antibodies were what were made in response to the antigens, then we end up with agglutination, these clumps being formed, and that's a problem. So there is going to be a picture on the quiz that looks like this. This is the exact picture, and it asks you to recognize what are the antibodies here? Where are the antibodies? And antibodies are these green little things with these Y-like structures that come out, these Y-shaped shaped structures that are the antibodies. So recognize that shape on the picture. Those are our antibodies. And it was by recognizing these antigens. The antigens are kind of little flags that we have on top of the cell that tells that I'm this kind of a cell, so this is me. And if it's not what it should be, it will then be recognized. We did the blood transfusion matching together in the class, so you just have to remember what you can give to who. And the quiz will ask you that can you give this type of blood to this type of a recipient. Big thing to remember, oh, I didn't mean to go ahead. Big thing to remember there is that remember that it's gonna, I'm trying to find a way to pause that. Remember that just use this, look at the letters, don't worry about the pluses and minuses. You just want to get the letters right. We're not worrying too much about Rh positive or negative. On this class, we're just focusing on the ABO system. Well, the next thing that we wanted to talk about is, or the quiz asks, it asks you to recognize, is this AB type of blood or is it O type of blood? Well, if you have antigens for A and B, it's gonna be AB type of blood. Universal acceptor is the AB blood type. So people whose blood type is AB, sorry, it moved ahead, I didn't mean it to. People who are blood type AB, they can receive blood from anyone. So they can accept blood from anyone at all. This is the least common blood type in the US. Blood type O instead has no antigens, A or B. Sorry, I don't know why it keeps jumping. I'll keep fighting against it. So blood type O didn't have any of those sprinkles. Since it doesn't have antigen A or B, it has antibodies against A and B. And blood type O is the universal, oh my goodness, is the universal. I'm gonna go and just pause this slide this way. It's the universal donor. So you can donate blood type O to anyone at all. And this is the most common blood type that we find in the US. So that's really the big thing. So you can drag these statements that is it A and B, AB blood type, or is it blood type O? Did you get those? Yes. Perfect. I'm just gonna move ahead and show the next one. So as other student was really worried that is there gonna be questions about Rh blood group, and really there isn't much. There's a couple of basic questions. So we remember that most of Americans are Rh positive. That means that they have a D antigen. The big thing to take away from the Rh blood groups is, and I didn't mean to move ahead. I'm gonna again do the same so it stays on still. The big thing to remember about the Rh blood groups is that there is no problem during the first pregnancy, even if the mother is different from the fetus or the embryo and then the baby. So if mother is negative, fetus is positive, that's not a problem during the first pregnancy because we have a placenta that keeps the circulations of the two separate, the mother from the child. However, what will happen is that during birth, blood of the baby and blood of the mother typically always ends up getting mixed, especially when the placenta separates. So there is going to be mixing of those bloods, and during this time, the mother can receive, if the child was different Rh group, the antigens from that, and it will produce antibodies against that. Well, the baby is gone at that point. It's already out of the mother's womb, so that's not going to be a problem. But if the mother gets pregnant again, and it's going to be the same situation, that mother has now antibodies against of the child's antigens of the Rh, it will actually end up attacking the baby, and the baby will be, there will be probably spontaneous miscarriage due to that, because mother's body thinks that that's a foreign thing that it needs to attack against. A couple of other things. We will use this next slide in the class tomorrow. So just what it looks like in terms of different blood types when you're testing. There will be 4 pictures in the quiz where it shows that, okay, we have the anti-A is the blue and anti-B is the yellow. Well, if you have clotting happening, and I don't know why this keeps moving. If you have clotting happening with A only, that's going to be type A. If you have clotting happening with B only, that's going to be type B. If you have clotting happening with both A and B, it's going to be blood type AB. And if you don't have clotting happening with either one, it's going to be blood type O. So as long as you're able to tell that apart on the pictures on this quiz, you've got 4 points on your pocket. That wherever the clots happen, that's the blood type. So just know that. Wherever the clots happen, that's your blood type. Blood disorders, these are kind of the last things that come up on the quiz. So you can, if you have any of these disorders, you will then respond to that by producing more red blood cells. So if you have sickle cell disease, you produce more red blood cells. If you have hemolysis, you produce more red blood cells. If you have hypoxia, you respond to that by producing more red blood cells. If you have anemia, again, you produce more red blood cells. And if you're bleeding, you have hemorrhage, you produce more red blood cells. So all of these are conditions where we would produce more erythrocytes in response to these conditions. So erythropoiesis would happen in response to these conditions. There is also a question, where would we see then again the white blood cells being produced, so leukopoiesis happening. Leukopenia refers to a condition where you have small amount of white blood cells. Well, if you have leukopenia, the response to that is to produce more white blood cells. So there are certain conditions where it will not have any effect on the red blood cell or white blood cell production. If you have this condition, it will not change the amount of it will not make us to make more of these kinds of cells. In leukemia, hematology, primary and secondary polycythemia, there will be no improvement whatsoever. And again, this is going to be a question where you have those headings, improvement with the red blood cells, improvement with the white blood cells, or no improvement with red blood cell or white blood cell. And you are asked to drag these blood disorders under these headings. And just give me a wave when you've gotten them all. There's no rush. I just want to make sure you get all of these for your notes. Okay, good. Thank you. A couple of other blood disorders that, again, there will be the name of the blood disorder, and then it asks you to match the description of that blood disorder with that name. We didn't have time to go through disorders in our review together, so I'm going through them now. So disseminated intravascular coagulation refers to clotting, where we have clotting happening all over the body in blood vessels where there's no damage to the blood vessel whatsoever. So there should not be clotting happening, but it's happening. That's disseminated intravascular coagulation. A condition that's known as infectious mononucleosis is when we have an infection of B lymphocytes, and this is an infection that's due to Epstein-Barr virus. So as long as you remember that Epstein-Barr goes with mononucleosis, infectious mononucleosis, you will be able to match the definition and the disease. You might have heard that someone might be septic. You might have a septic shock. You might go into sepsis. Septicemia relates to this. So septicemia is when we have bacteria in the bloodstream. Oh, I didn't mean to highlight it with such a dark color. So I'm just going to remove that. So if we have bacteria in the bloodstream, we're talking about septicemia. And that would not be great, of course. Thalassemia refers to deficiency or complete absence of alpha or beta hemoglobin. So if you don't have hemoglobin or there's very little hemoglobin present, that's known as a condition of thalassemia. And finally, if you have very low platelet count, we talk about you being thrombocytopenic. So thrombocytopenia is when you have small platelet count, anything below 10,000 per milliliter. So those are disorders that it asks you to match in the quiz with the name of the disorder and with the description of the disorder. And I think there was one more list of disorders that it asks you to match. So we're going to have a look through that when you're ready. There's no rush. Just give me a wave when you get them. Okay. This is the, oh, there's two more lists. I'm sorry. So red blood cell blood disorders. So I listed the what's asked in the quiz. Again, they give you the name of the disorder and then it asks you to match it with the description of it. If you have anemia that results from iron deficiency, that's caused by the loss of blood without replenising the iron that gets lost. So we're losing that iron while we're bleeding and we're not getting more extra iron, so we become anemic for iron. Pernicious anemia, when your stomach glands fail to produce intrinsic factor. And primary polycythemia is a cancer of erythropoietic line of red bone marrow. So we remember the red bone marrow was where the formed elements were produced. If we have cancer within the red bone marrow, we would end up with primary polycythemia. Secondary polycythemia can result actually from dehydration. So dehydration alone can be a cause of secondary polycythemia. Sickle cell disease, that's the one that we talked about in a class. So that was caused by a genetic mutation in a recessive allele, and that changed the hemoglobins, the protein that's produced for hemoglobin. And because the protein that gets produced by this one amino acid being wrong gets the wrong shape, you are left with red blood cells that are not the shape that they should be, but instead wrong shaped. And because they're wrong shaped, they're not able to transport oxygen as well. And final one that I have there is thalassemia. That's the deficiency or complete absence of alpha or beta hemoglobin. And we had it on the last slide. So because it gets asked twice, I wanted to also mention it twice. So you have every chance of seeing all of those. And again, don't worry, don't rush. You take those notes and give me a wave when you're finished. I know that it's a lot of conditions. I'm a little bit against of having this many conditions in your quiz because you are not yet going through the med school, but we use the same quizzes in every section of the course. The difference is that I just show you the answers for the quizzes. The others don't. And I really think that it's useful to get those points from the quizzes because they add up real fast on the final grade. I do, though, at the end of the semester, you can move your final grade up by 1 letter grade by doing a project. And it's not
That's
a
kind of a
hard
lot.
project. Yeah, it is. I'm generous. I'm like all about get my students to get good grades. No. So there's disorders of white blood cells. And I know it's a long list, but I think it's the last list of disorders. So I'm going to show you those. If you're taking notes, I'm going to take the time to go through them. So there's different kinds of leukemias. The general leukemia refers to cancer where the production of the leukocytes and their precursors is increased. So in leukemia, we have a high amount of leukocytes and all the precursors that we saw what leukocytes are made of. So high amount of white blood cells is associated with leukemia. There's 2 types of subgroups of leukemia. In an acute leukemia, that's sudden, that's very rapid in how it progresses, and it is within months that if it doesn't get treated, death will occur. In a chronic leukemia, this is much slower to develop, and it might be months that we don't even detect that there's something going on. So the difference between acute and chronic leukemia, the speed of how quickly we first see it, and the speed of how quickly it's progressing. If you have leukocytosis, you have a high amount of white blood cells. Leukopenia, instead, you have a low amount of white blood cells. So these are the things that we see when we do the blood works, take the blood sample, spin it, and analyze it. Those would be things that we're looking your white blood cell count. And based on that, we can say, is it normal? Or if it's high, we say that you've got leukocytosis. If it's low, we say you've got leukopenia. Sometimes we can have a leukemia type that's known as lymphoid leukemia. In lymphoid leukemia, your lymphocyte production is uncontrolled and very high. Same goes for the monocyte production. That is, again, extremely high. So lymphoid lymphocytes. Myeloid leukemia instead has the granulocyte production being extremely high. So it's just knowing the difference that myeloid has to do with granulocytes, lymphoid with lymphocytes and monocytes in terms of the types of leukemia that we might have. And I promise this is the last of the disorders list. You're going to be an expert of disorders after this.
Hopefully.
You'll be good, I promise. And remember, you can use your notes throughout this semester. There's no such thing as proctored exam in my class.
I'm good now.
Perfect. I'm going to show you a couple of last things that were in the quiz. Again, this kind of an activity where we're asking you to read a picture or be able to understand what you're being shown. So here we have what happens if you have hypoxemia. In hypoxemia, you have less than a normal amount of oxygen in blood. So there's usually a problem with breathing or circulation. So in response to that, your bone marrow will start to promote erythropoiesis. So your bone marrow promotes making new blood cells. And that's all you need to answer in that question. It just asks you to recognize that if you have hypoxemia, your bone marrow will make more red blood, any blood cells, any types of blood cells. There is a question about hemophilia. That was the bleeding disorder that I told that I have. So this is sex-linked and inherited from your parents. It is more common in males, so it gets inherited to males. There's 2 types. Hemophilia A is the classic hemophilia, which is characterized by the lack of factor VIII, and hemophilia type B, lack of factor IX. So just as long as you know that that's a sex-linked common in males. You're doing well. A couple of terms that are asked also, and I think this is the last one. Yes, this is the last one that I have from the quiz. Hematocrit tells us the percentage number of red blood cells per blood volume. Hemoglobin concentration tells us the concentration of hemoglobin per volume of the total red blood cell amount. And red blood cell count tells us the number of red blood cells in the blood. So those are, again, if you can match the term with the definition of it. And that's all you have to do. The practice anatomy, as before, you just do it. It's just pictures. And I wanted to remind there's tutoring available, but I'm also happy to tutor. And that's it. I know we stayed overtime, so I'm going to give you like loads of extra credit once we get the next chance that I can add them in. So I really appreciate it. And
It's
tomorrow
okay.
should be a good lab. We're going to do the blood typing, so it should be easy stuff. Is there anything I can help you? Do you have everything you need?
I think so. I already looked, or I tried to look through the slides for the practice anatomy, so I think it's all good.
Don't worry. Yeah, I'm sure you're going to be fine. Remember to use your textbook, your notes, and I really do appreciate all the work that you're putting into this class. I can definitely tell it, and I will make sure that you get recognized for it. You're working so hard.
Thank you. I think I realized I forgot to read the textbook last week, which would have, I don't know why. I forgot we just had a textbook. So I think this week it'll help looking through it.
Yeah, don't stress too much over it. The quiz questions, the textbook really helps to tackle those. But really, for the exam, it is only the things that I talk to you about. I don't ask anything else. But on the quiz, there's few questions because we want you to read a little bit of the textbook.
Okay.
But if you don't have
good.
any questions, I'm just going to wish very good night for you. But if you do have, I'm going to be here. I'm going to be available. But I just really want you to know that I so appreciate all your effort.
Okay, thank you.
Have a good night. Bye.
You too.
And that really concludes our review of this week's material for the quiz. And I hope that you have found this useful. And like I said, we've had quite a bit of challenges with the voice, with the WebEx in general today. So I will be sure to upload at least one version where we have a voice on this recording. And on that note, have a very good rest of the night.